Good morning, everybody. Can everybody hear me?
Yes.
Okay, I just want to make sure we've got a good link and everyone can hear before we get started. Okay. I'll do my best to kind of catch and read the messages in the chat. I can't promise I'll catch everyone, so I would ask if you do have a question, you can try to do the hand raise thing. It might be best just to unmute yourself and say, "Hey, I have a question." So I'll shut up and give you a chance to ask your question before we go on. So I want to take just a couple minutes and kind of introduce myself and the program and that kind of stuff. And then I'd like for each of y'all to go ahead and just briefly unmute yourself and just tell us a little bit about yourself. Basically, what are you doing in life right now? Are you a student? Are you a teacher? And kind of some of the same stuff I asked on the registration form. And then what are you hoping to get out of the course? So why don't we go ahead and just do the introductions first? I'll start. This is my name is Dr. John Thomas. I'm an associate professor of medicine here at the UTRGV School of Medicine. I'm in the Department of Human Genetics, and this is the mini course for the Center for Genomic Research. So the Center for Genomic Research is a group that was created out of a grant that we obtained through the NIH, partnering up with two other institutions, with the University of Hawaii and Case Western, to go ahead and establish a Center for Genomic Research that will help build genomics research capacity. And we're really trying to do this in two ways. One way is we're trying to expand the research capabilities and the discovery, the ability for researchers to go out there and do genetics research, maybe get some funding or support from our center. That's one of our focuses with this grant. And the other focus is we really wanted to try to enhance the size and the quality of the workforce that's available to participate in genomics research. Particularly in an area like down here in the Valley, we see a lot of the people that get educated here oftentimes take their degrees and leave, primarily because they're trying to advance their degree, they're trying to advance their education, and maybe what they're focused on is not going to be covered at an advanced level, perhaps at UTRGV, or if that topic is covered at UTRGV, there may not be enough companies or private corporations or work opportunities here in the Valley to be able to keep those people here. So we end up bleeding off and losing highly educated staff to other places. I've experienced this directly. I've been here since 2014, and I've had an externally funded research lab where I have students come in that are master's students. They come in and do a master's degree, do some research, publish a paper or two, and then they end up moving on to get a PhD, or maybe they go to medical school or something like that. But these people that are highly educated, that are highly trained, and my research focuses on immunology, virology, and using animal models to try to understand how viral diseases progress. These people have developed a very specific set of skills, and they have many times end up taking those skills and leaving the Valley and going other places, maybe to San Antonio or to Galveston or to Austin or even to Dallas, where more of these biotech companies and pharmaceutical research companies are headquartered, so they can use their degree and get a good job. And there's absolutely nothing wrong with that. One of the things that we would like to try to do with the Center for Genomic Research is to try to see if we can use an interest in genetics coupled with our approach to try to help educate people as to what some careers are in genetics, to see if we can expand the pool of people that are interested in doing genomic research, clinical scientific researchers at both the doctoral and the technical staff levels who can help perform this research. Maybe you're somebody that likes doing the research, but you don't want to get a PhD and be in charge of a lab, or you don't want to go to medical school and have to deal with all that. That's fine. You can still have a very good, productive career that'll pay you well, working in a field such as genetics. And so we really want to try to use this mini course, along with some other offerings that we have under this Center for Genomic Research grant, to try to push this out into the community, to let people know that this is a career path and an opportunity that's here locally. And I think that as we see the Valley continue to expand in the future economically, you're going to see more opportunities for doing this kind of research staying here locally. And so we want to try to get this information out there. And maybe some of y'all that are coming in are in a position where you're maybe working with high school students or maybe even middle school students, where you could maybe take some of this information, and we're going to share all the resources and the slides that we use for doing these talks at the end of the mini course. So you can then take this information back and maybe disseminate it out to people that you think might benefit from it. Again, our goal is to try to, create a pipeline if we can, to try to show people that, hey, there is an educational opportunity at UTRGV that's beyond doing something that UTRGV's maybe been more traditionally focused on, which is stuff like evolutionary biology, for example. We have a lot of really good biologists in the UTRGV College of Sciences that focus on the unique animals and plants that are down here. If that's not stuff that interests you, there might be other opportunities here that would. And so we want to kind of take this opportunity to try to show people what is available at UTRGV as well as around the world and the country. And our emphasis is going to be, with this mini course, genomic and genetic research. So that's kind of what the center's goal is. We have this mini course that we're going to run. We're going to do the mini course sessions here for the next 4 days. And then we're going to offer a repeat of this mini course in the middle of August, to try to give people that maybe couldn't attend this time, because this is being held at kind of a weird time. If you're a high school teacher, I know I have kids in high school right now and they're in school. So it's really difficult for a high school STEM teacher to duck out of class right now. So we're going to be offering this mini course 2 times a year. And then we'll make a decision here at the end of this year what we're going to do with the mini course moving forward in terms of the content. But as I said in the flyer, we've got a specific range of topics that we want to cover. The depth of coverage for these topics is probably going to be at about an undergraduate level of understanding. And again, this is not because people are dummies or anything like that, but we really want to try to make sure that we capture as many people as possible with a wide net. And some of y'all coming in probably haven't had genetics. There may be people coming in that haven't had any kind of immunology or any information or knowledge on bioinformatics. And so we really want to introduce these topics. And then again, hopefully you can take this information and share it with people that you think might benefit from it, so we can try to see if we can help drive people to a career in genetics research here at UTRGV. So let's go ahead and just kind of start from the top, I guess, and have people introduce themselves. I think we'll go ahead and start with you, Natalie, since you're at the top of my list.
Okay. Hi, I am a junior undergraduate student at UTRGV. I have a major in biology with a minor in rehab and a concentration in teaching.
Okay, great. Are you from the area?
I am. I'm from Brownsville.
Okay, awesome. Well, welcome to the class. Okay, Karina, why don't you go next? Karina, are you there? Okay, Louise, how about you?
I'm so sorry. Yes, I'm here.
Okay,
Okay, so I'm Corina Caballero. I'm a UTRGV student here in the Brownsville campus. I also work at the Career Center. So I want to follow like the STEM field, but I don't know like exactly where that fits in. So when I saw the flyer, I was like, this is a perfect opportunity for myself, and also in the path I'm in. I'm currently at the Career Center. I see students, and there's a lot of students who are like, oh, I want to do this kind of work, but I don't want to leave the Valley. So I'm also hoping to bring that back into the office, so the students that we see also know that there's a career for them here locally. Maybe they do have to move like to McAllen or Harlingen, but it's still not like a whole different state, where usually we only know about companies that are like out of state.
okay, awesome. Thank you. All right, Luis, you're up, man.
Hello, can you hear me?
Yes, we can.
Okay. Hi, my name is Luis Vicente Gutierrez. I am a biology instructor here at SIVCS, and I am mainly teaching the collegiate campuses, the high school campuses. So that's where I think this might help me deliver some of these more difficult topics to the high school students and see what I can learn from that.
Fantastic. Welcome to the class. Okay, Angelica?
Hello, can you hear me?
Yes, we can.
Okay, perfect. Uh, I am a lecturer in the biology department. I am based in Edinburgh, and Dr. Pettis did forward me this information since I am going to be working at the early colleges, the collegiate high schools. So that's the main demographic that I'm going to be working with for general biology and anatomy. But I think this is perfect opportunity to sort of introduce right to the younger generation of maybe some local research. You know, you made the point, right? You don't have to move all the way to Dallas or San Antonio to continue in your work. But I think this is a great opportunity for driving local career opportunities.
Fantastic. Thank you. Welcome to the class. And Christina?
Hello, my name is Christina Ortega. I teach undergraduate biology courses. I'm a lecturer, and I have a master's in biology. And I'm just really excited to learn more about the genomic tools and analysis that we have available. And it just would be helpful to have this knowledge to give back to the students and give insights on the different techniques and opportunities that are available.
Great. Thank you. Welcome to the class. Valeria?
Hi, can you hear me?
Yes, we can.
Hi, my name is Valeria Lopez, and I am an undergraduate junior at UTRGV. My major is in biology, and my minor is in business administration. I believe that relearning about this topic would really benefit students at UTRGV. I am very involved in clubs like the Pre-Medical Society and AMSA and other biology clubs, where I know that students that they join as freshmen don't really know where they want to eventually go or study. So I believe that knowing this knowledge and presenting this opportunity could be very good for them.
Great. Thank you. Welcome to the class. And I think Maya, you're the last one.
Can you hear me?
Yes, we can.
Hi, I'm a undergraduate student at UTRGV Brownsville, and well, I saw this as like a good opportunity to like um expand my career interests. I'm a biology major with a minor in psychology, and like I'm not set on anything, so I just wanted to like learn more about genetics and just like expand my opportunities.
All right, great. Thank you. Welcome to the class. And I have one person that showed up in person here. I'm not going to swing the camera to him, so you'll have to trust me. He's a handsome looking guy, but his name is Raul. So Raul, tell us a little bit about yourself.
So I'm Raul Ayala. I'm actually taking a postdoc in biology right now. My plan is to apply for master's in molecular biology or a PhD in human genetics. I think to know about the genes, that's the foundation for us to start helping or healing our people, especially in the area, you know. And that's why I'm here to know more, to have more knowledge. Because I think if we don't have knowledge, it's easier for people to become more irresponsible, and that's where we create all this disease. So I think this is the foundation to start healing our people.
All right. Well, thank you, man. Welcome to class. So I'll go ahead and just talk real briefly about myself. I gave my name. I've from Spicewood, Texas originally. It's a little town that's outside of Austin, about 30 miles outside of Austin on the shores of Lake Travis. My mom is from the Valley. My mom actually went to Homer Hanna High School, and then her senior year, my grandparents bought a house in San Antonio, so she actually graduated there. But I've been coming down to the Valley since the early 70s. I live about a mile from where my grandparents owned a house down here for about 70 years. So been down here in the Valley probably off and on, probably longer than most of y'all been alive. My wife is from Brownsville originally. And I got a position at UTRGV in 2014. It was actually UTPA that last 3 months of being UTPA. And then we formally became UTRGV. I think it was in December or so, or August of 2014. So I've been here since then. I've kind of gone back and forth between working at the College of Sciences and working at the medical school. In 2020, I moved over to the medical school and the Department of Human Genetics full time. And during the COVID pandemic, I ran the medical school's clinical diagnostic lab for doing all of the COVID testing, as well as all the testing for routine chemistry and hematology and that kind of stuff. In 2023, I came back to the department full time, and my research focus is on my training is in classical virology and immunology. And so a lot of my research is focused on using small animal models to try to understand how viruses and bacteria cause disease. And I've also spent a lot of time in vaccine design and drug research. Prior to coming to UTRGV, I was a postdoc fellow at the CDC for 18 months. I worked at UT Southwestern as a postdoctoral fellow for 2 years, working with hemorrhagic fever viruses. And then I spent 3 years working at a Fortune 500 pharmaceutical company in Iowa, making vaccines for dogs and cats. So I've been here since 2014. I, God willing, plan on finishing out my career here. I live in McAllen. I've got an office in McAllen. I have some lab space on the Edinburgh campus, and I have some space down here in Brownsville. So I often kind of bounce back between the 3 campuses. I've got 3 daughters. One of them is a senior at UTRGV. She's working on her undergraduate degree in computer animation and 2D art design. And then I've got a 19-year-old that's starting in the fall, working on a degree in cybersecurity. And then I have a 16-year-old who's a sophomore, I guess. Yeah, she's a sophomore in McAllen. So I have all of my children and my family's here in the Valley. So one of the reasons we decided to come to the Valley after having been everywhere else in the country was because we believe in the people here in the Valley. And that's probably the best resource that the Valley has are the people here. And so we wanted to come here and try to see what we could do to kind of help educate and provide opportunities for those people here in the Valley that might have an interest in science and research, but just don't know how to get it started. Or as many of you have commented, you really don't have those opportunities here in the Valley. I do think that that's something that's going to change here in the near future. I saw a news story last night. There's a major liquid natural gas company that's going to be building a brand new liquid gas terminal in Brownsville. And that gas terminal is probably going to create 2,000 to 3,000 really good paying jobs for, people that want to go out there and work in the gas industry, in the liquid natural gas industry. So I know from my perspective, I live in McAllen, and every time you drive up and down 10th Street or you look around, there's a new building going up. There's a new apartment complex. There's a new shopping mall. The Valley is really, I think, starting to go through a renaissance of development. In my travels, living all over Texas, working at every different UT school that there is in every part of the state, the Valley's probably been one of the last areas in the state, not counting far west Texas, to really get a lot of money pumped into it for economic development. But you see that happening now, and it is good and bad to some levels. I mean, you're seeing a lot of new people come in, and that's always going to be sometimes disruptive because they have to kind of get acclimated to the culture. Every part of Texas is different. South Texas is completely different than Central Texas and North Texas and East Texas. And so it's almost like their own little state, and you kind of have to get adjusted to the culture there. But I see a lot of economic development happening. I tell my kids all the time, you got to look around, because in 2 years, those empty fields are going to be a shopping center. That's going to be a business park. There'll be a new hospital over there. I think we've had 3 new hospitals built in the Edinburgh McAllen area in the last 7 years. UTRGV is building a cancer center right now that's scheduled to open sometime here in late 2025. So there's a lot of development and work and things that are happening here in the Valley. And I've seen several large companies that have made commitments to move their operations from places like California, and they're looking to come to Brownsville. I think Elon Musk moving SpaceX here has had a lot to do with that. And I think it's helped kind of maybe demonstrate that, hey, there is a capable workforce that's here in the Valley that is very dependable and reliable. If they can get the education that they need to help qualify for some of these positions, then, this will be great for everybody. I don't know how many of y'all know much about SpaceX or some of the people that work out there, but the top people that are running the SpaceX side out there are from Brownsville. They're from the Brownsville area. They're local people. These aren't people that were brought in from California or, some other place. So Elon Musk has done a really good job of recruiting and finding really good talent locally. And I think that's demonstrated to other big companies that, hey, there's a good workforce that's here. And so that's part of the reason why we're trying to lean into that resource here as well by telling people, hey, there's another path that you can look at and examine for working in genetics and biomedical research in general that's done here at UTRGV that a lot of people may not know about. The Department for Human Genetics that's here on the UTRGV Brownsville campus is one of the top genetic research groups in the world, certainly one of the top groups in the country, if not the world. This group has collected over $100 million in research grants in the last 10 years. That's an incredible record of funding. And we've put out a massive amount of publications and made some really cutting edge discoveries as a result of it. And so we want to try to take that knowledge and disperse it out to try to help expand career opportunities and educate the workforce pool that's down here on opportunities like this that exist. And I think it'll be a great benefit for everyone that can participate, not just in courses like this, but looking at these other companies that are coming in. They're going to be looking for educated employees that can come in and fill mid-level technical jobs, where maybe you're not going to be the person that's going to be the boss of the plant. But if you're someone that can be happy with getting a mid-level job that's going to have a stable salary, a good pay, good benefits, and you get to stay locally, for a lot of people, that checks most of the boxes that you have for a career. So I'm excited to have everybody here. I think this is great, and we'll go ahead and get started. So thanks everybody for coming in. And our first lecture today is going to be on I'm going to do a basic introduction on genetics, and we will talk about genetics and how it's used in forensic science. So I don't know if many of y'all are aware of this or not, but at Sam Houston State University, which is a small private-ish college that's located outside of Houston, it's about probably 45 miles west of Houston. It's in Huntsville, where the state prison is at. My dad worked as a prison guard there for about 10 years. But in Huntsville, at Sam Houston State University, they have a body farm. And a body farm is essentially a wooded location where they take human cadavers out and they bury them. And they do this so they can study how a body decomposes over time in a natural environment. And they do this to help them when they recover skeletal or human remains out in the woods, to try to make an approximation as to how long that body's been in the woods. And they can tell by looking at what kind of bacteria are growing, what are some of the maybe worms or snails or fungus or grass that grows around that body. Because of the substances in the body that's released as it decomposes, it alters the bacteria and the fungal and the environment for growth around the body. And so they do a lot of forensic research up there, really focusing on how bodies decompose and that kind of stuff. So a lot of the knowledge that we've actually put together, when you're watching CSI or something like that, and the guy comes out and they look at the suitcase that they dug up that had a body in it for 10 years, it was buried in the forest somewhere. A lot of the guesses that they can take, the educated guesses and predictions that they take and make, are based off of the work that's done at this body farm. So I've always thought that DNA and forensic research, particularly the genetic aspect, is really interesting. So we're going to talk about that today, and we're going to do a brief introduction into genetics itself. So again, as I said at the beginning, if y'all have a question, just go ahead and say so. Probably going to be the easiest way to catch it. If at any point you can't hear me, or if you think I'm speaking too fast, you need me to repeat something, please ask me as well. I've got a somewhat severe accent, but I'm doing my best to try to control it and speak like a regular white person. So if it gets too crazy, let me know, but we'll go ahead and get started. So obviously, when we're talking about forensic science, particularly looking at forensic DNA, many times this is being done to try to identify and link somebody to an event. So perhaps you're trying to link a suspect to a crime scene, for example. I'm not sure how many of y'all were alive in the mid-90s when we had the O.J. Simpson trial. O.J. Simpson was a very famous football player. He's in the Hall of Fame. His wife and her boyfriend were found brutally murdered, stabbed to death in their home in Malibu. and OJ was the prime suspect in this case. There was a lot of forensic evidence that was recovered there, and they used this forensic evidence to try to tie back to OJ. In some respects, they were successful. The forensic evidence did provide connections between OJ Simpson and the crime scene, but ultimately OJ was found not guilty because the way the crime scene evidence was handled after it was collected allowed for his attorneys, and he had very good attorneys, to go in there and make arguments against the integrity of the crime scene. So, for example, they collected bloody socks. They collected the bloody socks on a Friday night and then left them in the trunk of the police car all weekend. And this was in July in California. So after something has sat in a trunk in your car for the weekend, if you left a bag of hamburger from HEB in your trunk over the weekend, you're probably not going to eat that stuff on Monday because it's going to have gone rotten and nasty. And so the same arguments were used against OJ Simpson's prosecution for the DNA evidence that was collected, and so he was largely exonerated from the case. So DNA is used to try to link someone to a crime scene. It's also been used to identify missing or dead people. Obviously, everyone probably knows about Saddam Hussein and his sons Qusay and Qusay. DNA evidence was used to positively identify them when we got a hold of them in Iraq in 2005. When Osama bin Laden was killed, DNA was used to confirm that it was him after they killed him. So DNA is often used, and we probably see it all the time on TV. DNA has been used to identify missing people. They'll find somebody's body out in the woods, or maybe someone's foot or something washes up on shore, and we're able to take those remains and extract DNA from them, identify them. We've also been able to do this with soldiers that have gone missing in action. So there's a foundation out there that travels around the world, and they investigate old scenes of battles, down planes from World War II or from Vietnam, from other conflicts. They'll go out there and recover those remains and then bring them back and do DNA sequencing. And then they can actually, based on sequence information that they get from the descendants or the relatives of this man or woman, they can then positively identify the body. And then we can also use forensic data, DNA data, to link a suspect to an event. The best example I could think of recently that people may have heard of was the Bill Clinton Monica Lewinsky scandal in the mid-90s. Monica Lewinsky was a 20-year-old intern that worked for President Clinton. They had some kind of an affair, and there was biological evidence was left on one of the dresses that she wore. This became the blue dress event, and she saved the blue dress, and the blue dress was then turned over to the FBI, and the FBI was able to isolate DNA off the dress, and the DNA was connected to President Clinton. So overall, for applications, DNA has become a very, very powerful tool in forensic science. In many fields nowadays, but we're really going to focus on forensic science today. So because of some of the characteristics of DNA, DNA can be used as a very unique marker to identify back to a single individual. Your DNA and the structure of your DNA and the layout of your genes and how they are formatted is going to be as unique to each individual as fingerprints are. And so sometimes, as you may have seen, they will sometimes solve a murder case that was 40 or 50 years ago by having the descendants or the relatives of the victim submit DNA samples so they can build a DNA profile of who the victim was, and then they take a DNA sample from the crime scene and compare it to all those people and then eliminate everybody from the equation. So then they say, okay, the DNA sample that we got from the crime scene has a small piece of DNA that's not connected to any of these relatives. So this is our suspect. This is who we're looking for. And it's that uniqueness within the DNA molecule that has allowed us to use DNA to really be able to connect and tie back to individuals. So let's talk a little bit about what DNA is, how DNA is composed. For y'all that have already had genetics, this is not going to be a deep dive into DNA itself. I'm not going to go over some of the really granular details about DNA and genetics, but we will take a kind of a 10,000 foot view so everyone has an understanding of what DNA is and what some of the terminology is as we move forward. So DNA stands for deoxyribonucleic acid. Some of this stuff y'all have probably heard before. And the DNA molecule is contained in virtually every cell of our body. There are some exceptions. There are some cells that don't maintain DNA, but by and large, the vast majority of our cells in our body have nuclear genetic material inside of them that are called chromosomes. Male chromosomes are one X and one Y. Female chromosomes are going to be two X chromosomes, an X and an X. Arranged on these chromosomes are functional units that we refer to as genes, as indicated on the graph up here. And then within each gene, we have, we call this entire thing DNA. The gene is basically the fundamental unit of heredity. These are the instructions or the blueprints for the cells to make proteins. And these proteins are what are going to determine what color our hair is, how tall we are, whether we have skinny fingers or fat fingers, how long our fingers are. Virtually every one of our physical characteristics are connected to DNA and the proteins that are expressed from the genes located within DNA.
Excuse me, Dr. Thomas, you mentioned a graph, but we're looking at the mission statement.
Oh, I'm so sorry. Thank you for saying that.
No problem.
I did not share the right, I moved on and didn't even share the right things. Thank you for saying that. Was there no one gonna say anything else? Y'all just gonna let me run?
Oh, I put it in the chat.
Oh, okay. Yeah, if you can, try to let me know verbally if you can, because I'm not sure if I'll catch all of the comments in the chat. So, okay. All right, let's try this again. Okay. Can everyone see the presentation now?
Yes.
Yes.
Okay, what is DNA? Okay, thank you, Luis. I apologize for that. Everyone else gets -5 points. I'm just kidding. Okay, so here's the chromosome that's located within our bodies. XY for female, or XY for male, XX for female. And on these chromosomes are made up of basically the material that we call DNA. And within the DNA is a functional unit that we refer to as a gene. And the gene is basically going to contain the instructions that I talked about that's going to encode a protein, and this protein will then go out and perform some specific function. It will determine your hair color, your eye color, how long your fingers are going to be, how tall you're going to be, what color is your skin going to be, you know, all those kinds of things. So if we look within a gene itself and we try to identify exactly what makes up a gene, we basically have nucleotides, DNA nucleotides, that are going to be linked to each other through we call a phosphodiester bond that's going to link different little nitrogenous bases together. So this is kind of the chemical structure, so everyone can be fancy and look at the chemical structure. But if we look at this from more of a cartoon perspective, basically what we're going to see is we're going to see 4 different types of bases that we refer to as A's, T's, C's, and G's. And these are technically known as adenine, thymine, guanine, and cytosine. These are the 4 bases that make up a DNA molecule, that make up the gene, that make up the DNA molecule. And these 4 bases, the A, the T, the C's, and the G's, are going to be aligned in different combinations. And these different combinations are going to come together to form the DNA molecule. We know that the DNA molecule is a double helix. This was a structure that was discovered in 1953. Watson and Crick get the technical credit for it. They won the Nobel Prize. But this structure was actually discovered by a woman who was an X-ray crystallographer at Oxford University. She was actually the one that did the crystalline structure work that got the 3D, that got the 3-dimensional double helix model. And so she was at the time working as a technician in the lab. And so they essentially took most of the credit for the discovery. Not that they didn't make some work on the discovery itself, but there's actually a woman that did a lot of the technical work that doesn't usually get a lot of the credit. She certainly wasn't given any credit for the Nobel Prize. And the reason that Nobel uses for this is because she died from radiation poisoning, sadly enough, before the Nobel Prize was awarded. And Nobel has a rule that they don't give out Nobel Prizes to dead people. So if you make some awesome discovery and you cure cancer, you better live long enough to make sure that you get the Nobel Prize, because even if you cured cancer or HIV and you end up passing away, they won't give you the Nobel Prize. But Watson and Crick are the ones that we formally credit with the discovery of the double helix structure of the DNA molecule. And you're always going to have these A's and T's and C's and G's. And the way that this generally functions is that the A's are always going to pair with the T's, and the C's will always pair with the G's. And we'll talk about more about this and look at this here in just a moment. But these A's and T's that are being held together up here, and the C's and the G's, these bases that are being stuck together, are held together by very weak hydrogen bonds. So if you think about the double helix molecule itself, running as the double helix here, you have these A's and T's and C's and G's that are being held together, because again, remember, this is a double helix. This is two strands running together that start to twist around. Let's see if we have a better picture of the double helix. So here's a little bit of the double helix right here. So these are A's stuck with T's and C's stuck with G's. And so as these things run up and down the double helix, these weak bonds are going to be formed between the A's and the T's. And this is what helps hold the DNA molecule together. This is oftentimes referred to as forming the rungs of the DNA ladder, because if you look at this, this kind of looks like rungs on a ladder. If you were going to step up this may not be the kind of ladder you want to step on to get on your roof, with these things being all crazy, but you're always going to have A pairing with T and C's pairing with G's. You will never have A's and G's pairing together or C's and T's pairing together. There's several reasons why this won't happen, but if you think about this from a real perspective, we're looking at a cartoon that's just showing us these two bases stuck together. But in reality, there are many little pieces sticking off this DNA molecule. There's going to be other little proteins, little pieces of glucose, other little pieces are going to be sticking off this thing, and they will physically block the interactions with the wrong base. These things have to kind of fit together like puzzle pieces. And so if you try to jam two puzzle pieces together, you can maybe force them together just by pushing them, but when you let go, they're going to come apart again. And so the DNA backbone is associated in such a way to where A's will always pair with T's and C's will always pair with G's. But we don't want this pairing to be super strong, because when we copy a DNA molecule, we have to break these hydrogen bonds to get these two strands away from each other when we copy DNA. So we want to have enough of a bond to hold the A's and T's and C's and G's together, but we don't want it to be a super glue bond that can never be broken. We want it to be a bond that can be broken with just a little bit of energy, and that's why it's a hydrogen bond. So we have the A's and T's that are holding, and the C's and G's that are complexing with each other, and this in large part helps to kind of stabilize the DNA molecule and hold it together. So when we want to replicate DNA, go away. When we want to replicate a single strand of DNA, what's going to happen is that the base that's complementary to the base on the strand is what is going to be generated. So A links up with T. So you're going to have, if you have an A on this strand, then the other strand is going to have a T. If there's a T, then this one's going to have an A. The same with the G's and the C's. So when a single strand of, when DNA is replicated, the double helix must first be broken apart into a single strand. And then the single strand is going to be paired with a new DNA strand that's going to have bases that match and complement the bases over here. So in many respects, you can think of this as the instructions. Really good IKEA furniture instructions that are super clear and you never grab the wrong screw. So you have this strand as the strand that is the parental strand. That's going to be the instruction strand. The strand that's going to tell you what needs to be brought over. And so the strand that's brought over is going to have a T that complements this A, an A that complements this T, because remember, A's and T's go together and C's and G's go together. So if you have an A on this strand, this other base has to be a T, so these can link up together. So this is how DNA is going to be replicated. Now, we're seeing just a small piece right here, but a DNA molecule, the average DNA molecule in a human being is about 3 billion of these letters. You could think of it 3 billion letters written down on a line. If you had to copy 3 billion letters of A, T, C, and G in this kind of a pattern over and over and over, you'd probably make a lot of mistakes. And so there are certain rules that the cells and the body follows with respects to how DNA molecules are copied to make this process as foolproof as possible. So to kind of give you an example, for every 1 million bases that are copied, the body makes one mistake. So if you picture writing down ATAGC, writing this out 1 million times until you've written down 1 million letters, if you were the body doing this, you'd make one mistake. Many of us would probably make 100 mistakes or more. But this process is very tightly regulated in the body because, and we'll talk about this in a moment, if the wrong base is written down, then you're going to get a change in the protein, which is going to cause a mutation, which may not be beneficial for you. So this is a very tightly regulated process in the body. And within our body, within the human body, and most of and all of the higher animals, the eukaryotes is what we refer to these as. This process is able to undergo a phenomenon known as proofreading, which basically means once you write down and the body writes down all of the bases, it can then go back and check its work. So just as you might tell your students in class, or you may have your professor tell you, hey, before you turn in your assignment, check your work and make sure that it's right. The body does the same thing. It will go back and check and make sure, hey, this was an A. Did we make a T? This was a T. Is this an A? And if it finds a mistake or an error, then the body can correct that mistake, which is very good for us because the DNA in our body is being copied hundreds of times a day. Every time the cells in your body divide, you get a scratch on your skin. You need to make new skin cells. Those skin cells, every one of them has a 3 billion base pair of DNA molecule inside of it. When you make new skin cells to fix your scratch or your sunburn, that DNA within that skin cell has to be copied again, and it needs to be perfect. Because if you get a mutation, then you could have problems with your skin. So the body is very, very good at replicating DNA almost perfectly. We make one mistake for every 1 million bases that we copy, and then we can go back and correct that one mistake. This is something that bacteria and viruses cannot do. If a virus or a bacteria copies its DNA and it makes a mistake, so be it. It lives with the mistake. And maybe the mistake gives the virus or bacteria a benefit. Maybe it ends up hurting it. But bacteria and viruses don't have the ability to fix mistakes in their DNA. The eukaryotic organisms, such as plants, animals, insects, yeast, parasites, we can go back and make corrections to our DNA molecule. So this is a very tightly controlled process, engineered and designed to be perfect. Why does it have to be a perfect process? Because all of these little letters are going to encode for proteins. So each one of these bases are going to encode for an amino acid. And we're going to talk about this right here. So there are 20 amino acids that we know of. And arranging the amino acids in different combinations gives us different proteins. We can produce tens of thousands of different proteins from the 20 amino acids that we know about. And each protein that's produced can perform a different function. There's a very common phrase in genetics and in proteomics, which is the study of proteins, which is shape determines function. When proteins are produced, they have a unique three-dimensional shape. And that unique three-dimensional shape determines what the function of the protein is going to be. If we change the genetic code and we change the amino acid in a given protein, then we're going to change the shape of the protein. And if we change the shape of a protein, the protein loses that function, because for proteins, the protein shape determines the protein's function. This is again why the process of DNA replication is so tightly regulated in many organisms, because if the wrong proteins are made, that organism is going to die. So the DNA replication process has got to be very, very perfect as we can make it. So we have these different amino acids, about 20 different amino acids that we know of, that can be arranged in different sequences that can make about 100,000 different proteins, each with their own unique shape, which then means they're all going to have their own unique function. So how is this done? So we're going to go through this part right now. So DNA has to first be changed into, when it's going to be copied, has to first be changed into RNA, which is ribonucleic acids. It's very similar to the DNA molecule. And I'm assuming everyone's probably heard of DNA and RNA at this point. The big difference between DNA and RNA is that there is no A in the RNA molecule. So we have A, T, C's, and G's as the 4 nucleotides in DNA. In RNA, there is no A. The A is instead there's no T. There's no thymine in the RNA molecule. There's no T. So the T is replaced with a U. So in RNA, A is going to bind with U because there is no T. And this is how you can easily determine when you look at a sequence of genetic material, whether it's DNA or RNA. If you see U's in the genetic sequence, then you know that it's RNA. If you see T's, then you know that it's DNA. And then each amino acid, how does the body know which amino acid to bring? Because it's given the instructions from the triplet codons on the RNA that tells it which amino acid to bring over. So what does that mean? So I've got a nice little picture right here. So this is our DNA sequence, okay? A, T, A, G, C, C, A, C, C, A. So in DNA, we have A's and we have T's and G's and C's. When this gets changed into RNA to form a protein, this is what we end up getting instead. Now, when A is going to be matched up, remember, there's no T in RNA. So now the T's are U's. So you still have A's in the RNA molecule. Okay, so if you have a T in the DNA, you're going to get an A over here. But if there's an A on the DNA molecule, the RNA molecule is going to have a U. And what we do is the body looks at these letters in groups of 3. So these 3 here, and then these 3 here, and these 3 here. And each one of these groups of 3 are referred to as a codon, C-O-D-O-N. And this codon is then going to tell the body which amino acid to bring over to make the protein. So we have our DNA template strand up on the top, and on the bottom, we have the RNA strand that's now been transcribed. So each group of these 3 nucleotide bases is referred to as a codon, and it's read in order from left to right, just as we read normally. And this tells the body which amino acid to bring over as we make our new protein. So now we have all of our codons brought over. We have our transcribed piece of RNA. Now the body's ready to look at these letters in groups of 3 and say, okay, UAU. What do I bring over for UAU? Well, we have a table that tells us, okay, here's U, here's UAU. Okay, UAU means bring over the amino acid tyrosine. So the body would then grab in the cell, grab some tyrosine and bring it over to start making the protein. Well, what was next? The next one is CGG. Okay, well, which is next for CGG? Where's CGG? Well, here's CGG, that's arginine. So using this codon, using this triplet codon, the body is able to bring over the amino acid that corresponds to each one of these codons. So remember, codon is groups of 3. So this group of 3 is going to be tyrosine. This group of three was, I think, adenine. And so the body's going to read these groups of three and then bring over the appropriate amino acid. And it's going to hook these amino acids up together. And as you add amino acids, you're making the protein. So these are the bases that we know of so far in the codon. So here are your positions. And then if you look here, you'll see that the last letter in each one of these codons that's coded green, the reason that it's coded green is that the body has built, again, the design of DNA replication is very, very elegant. And it's designed to be flexible for an organism to be able to function and survive, right? You have to have a little bit of flexibility. We need this to be as perfect as possible. We need there to be very few errors. But even if you can only have an error of 1 in a million, when you're doing this billions of times a day over 70 or 80 years, the chances are good that you're going to have some errors, that the body's going to make some mistakes. One mechanism that the body uses to address these errors is this third base, this green letter, the third codon here at the very end, has some flexibility in what it could be to code for the same amino acid. What do I mean by that? So if we look right here at UUA, this codon in groups of three codes for leucine, but UUG can also code for leucine. So the goal when DNA is being transcribed is to ultimately make a functional protein. So at the end of the day, the most important thing is the amino acid that's being brought into the protein. So by having the ability for two different codons in the RNA molecule coming from the DNA molecule to encode for leucine, this gives us some flexibility. Maybe when we were making this copy, we made our body made a mistake and we didn't get the right amino acid over. Well, or as long as we have two opportunities to make leucine, because this could be UUA or UUG. We call this flexibility in the third position the wobble hypothesis. And basically all this means is that, hey, you can wobble, you can flex a little bit with this third position, this third codon position here, and still get the same amino acid. Look right here. This is CU for the first two letters. The third letter could be basically anything. Remember, in RNA, a U is a T. So if you want to make leucine and you've got C and a U, any one of these three bases at the end could encode for leucine. For isoleucine, you have a lot of flexibility. Look at threonine. Again, a lot of these amino acids, as you look at them, serine, alanine, valine, any one of these letters at the end of this group of three can give you the same amino acid. So the body has developed this kind of redundancy where if you maybe do have a mistake and the DNA is copied, you still have a chance to get the right protein made by getting the right amino acid brought over. So let's talk about this a little bit more. So here we have an RNA sequence. UAU encodes for tyrosine. And we can go back and see right up here on our little code, there's UAU, there's tyrosine. So this is called the codon table. And so we have UAU encoding for tyrosine, CGG gives us arginine, leucine, valine. These are the amino acids that are being brought over to build our new protein. And let's say this is a protein that's going to be used to grow new hair. No one, well, I shouldn't say no one wants to be bald, but for those of us that are maybe losing a little bit of hair, would like to try to keep some, you want this protein to be made properly. So you're happy that you get the chance to make leucine a couple times because maybe instead of getting UUG, this came over as UUC or UUA. Well, that's okay because if we look at leucine, we see that we have the opportunity to bring that back up. Now you'll also see on this codon table some codons that say stop. And you have a codon over here that says start. There are certain codons that are used in proteins to say, this is the beginning of the protein, this is the end of the protein. Because we need to have some way to eventually stop making the protein. And the way that this is done is by use of these stop codons. So in every protein that we've looked at that's been made, we find a start codon. It's usually going to be AUG, which encodes for methionine. This is almost always the first amino acid in any new protein that's made. And then the last couple codons are going to be either UAA or UAG, and this tells the body, stop making the protein. So every protein is going to have a beginning and an end, and then it's up to the body using the RNA molecule that's been made from the DNA strand to tell us what to put in there next. Is this kind of clear so far? Does everyone kind of understand this so far? Any questions on where we're at so far?
No, all clear for me.
I'm sorry?
I said no, you were very
Okay.
clear.
Okay, good. So then here's our protein amino acid sequence that we get. And if we make a single change in one of the bases, we did have UUG coding for leucine, but if a change gets made, now it's a completely different amino acid. Once we start to change the amino acids again, when we're looking at this as a cartoon for the purpose of understanding and learning. But in reality, this thing is not a freaking straight line. It's a twisted, folded mess of proteins that are inside of a solution in our cells, in our bodies, that's got salt and calcium and all kinds of other things floating around. So this protein has to take on a very specific three-dimensional shape in order to be able to have specific function. If the amino acid gets changed from leucine to phenylalanine, this is probably going to change the three-dimensional shape of the protein, which means that that protein that was going to be used to grow hair, now it's going to be used to make your armpits sweat more. It's going to have a different function. And this is one of the reasons why the body tries its very best to make sure that no mutations occur in the DNA molecule, because if you get a mutation in the DNA molecule, you're going to get a mutation in the protein, and there's a good chance, not 100%, but there's a good chance that you'll change the function of the protein. So does all of the DNA that we carry code for proteins? And the general answer is no. Most of it does. The part that we have traditionally been interested in, or that we focused on for the last 20 or 30 years, we really got good at sequencing DNA probably about 30 years ago. DNA sequencing really started to kind of come into its own in the late 90s, early 2000s. This is when we had the Human Genome Project in 1996 or 97, where we first sequenced the human genome. It took them about 3 years, and we got a chance to kind of look at what's in the DNA molecule. And our initial focus has been on the DNA that encodes for proteins, because we just spent 20 minutes talking about how important proteins are. And you got to have functioning proteins shaped in the proper way to make your hair grow, to make your armpits sweat, to help you digest, to help you copy more DNA, to help repair wounds and injuries. So we've been focused traditionally on what is the protein generating part of DNA. But we're now, as we've spent more time studying the DNA molecule, we understand that the part that makes up proteins is only about 5% of the DNA molecule in humans. There's another 95% that we don't really understand what it is. It's been traditionally referred to as junk DNA or filler DNA, because we didn't kind of have a good word for it, and because we initially kind of assumed, hey, the important part is what makes proteins. Who cares about everything else? We're now beginning to understand that 95% of DNA that doesn't encode for proteins has a lot of useful information in it, and carries out a lot of functions in terms of regulating how proteins are made and how other processes function in the body. And we're still in the process right now of sequencing and trying to understand what this 95% of DNA that we refer to as junk DNA carries with it. But we have some, of course, because it's science, we had to come up with fancy words to describe these things. And so the part of your DNA that actually codes for functional proteins, we refer to these as exons. The way that I remember this is that exons expressed, proteins are expressed. So that's the useful part, quote unquote, of DNA, are the exons. This is the part that codes for proteins. The other part that doesn't code for proteins, we refer to this as the introns. So the exons are expressed, the introns are inside, they're not expressed. That's how I always remember this when I was a student. So we've got about 5% of the DNA that's encoding for proteins, but we've got this other 95% that doesn't. But that other 95% carries a lot of uniqueness inside of it that helps us distinguish individual DNA molecules from person to person. And so some of the early DNA techniques that we use to try to identify DNA focused on looking at this junk or this filler DNA and cutting it with enzymes. For those of y'all that don't know or maybe have forgotten from basic biology, enzymes are proteins that are in the body whose job it is to go around and cut other biological molecules. Some enzymes will cut other proteins. Some enzymes cut DNA. Some enzymes cut themselves or cut other enzymes. But we've identified a pretty big library of enzymes, and we know where they cut within a DNA molecule, for example. So in the early 80s and 90s, I started doing this kind of work in the mid 90s. And at that time, what we would do is we would extract DNA, and then we would take enzymes and treat the DNA with these enzymes, and the enzymes would cut the DNA into different sized pieces. And then you can take these different sized pieces, and if you put them under a very weak electrical field, you can push them across what's called agarose, which is basically scientific jello. It's a very squishy jello type substance that will trap DNA molecules based on their size. And then if you apply electricity to this scientific jello, this agarose, with DNA on it, depending on the size of the DNA, it will move different distances. And so what we can then do is take a piece of DNA, cut it with enzymes, and then we can take the cut enzyme treated DNA and run it on over this agarose gel under an electrical field, and you're going to see the different size fragments move at a different speed. Basically, the smaller pieces move really fast, and the big pieces move really slow. So the smaller a DNA molecule is, the faster it's going to move under an electrical field. The larger it is, the slower it's going to move. So what we would do in the 90s is we would take a piece of DNA, cut it with enzymes, run it on the gel, and then you would get this banding pattern. And then we would refer to these things as restriction fragment length polymorphisms. Again, we love to give big important names to things in science, so it's difficult to remember, and we can make sure students have to study for tests, and we can mark them wrong when they don't spell polymorphic right. But basically, these are cut little pieces of DNA. So if I take your DNA and I cut it with some enzymes and I run your DNA on a gel, and then I have a sample of DNA from a crime scene that we think might be yours, and I take that and I cut it with enzymes and run it on the gel, and I compare the two gel patterns, I would hope if it's your DNA from the crime scene, they should match. They should match up perfectly, because I took your DNA that I know is from you, I took it from your hairbrush, and I got DNA from your hair, and I cut it with these enzymes, and I got all these different sized pieces that are going to be unique to you, because these sites in the DNA molecule where the enzymes can cut, the space in between them is going to be different sizes, because we're all different people. Raul here, his C fragments might be bigger than mine. So you can, because this is going to be unique to each individual, we can use these little pieces of fragments of DNA that we cut with restriction enzymes. That's why we call it restriction fragments. We can use the size or length of these things to identify unique individuals, therefore identify unique DNA patterns. So this was kind of the first attempt at trying to use DNA to identify people, and this is referred to as DNA fingerprinting. This is not a technique that's done anymore, partly because it's a huge pain in the ass to do this. It's very prone to error, and it's not very specific. If you go into court arguing that, hey, these two DNA bands look the same because I cut them with an enzyme and I know it's from this guy or this girl because their patterns match, that's a pretty easy argument to attack from a legal perspective, because you can always say, well, hey, there's 6 billion people on the planet. How do you know that there's not somebody out there that has the very same or a very similar pattern? So this is useful to an extent, but this isn't really something that you could probably stake your career on if you're trying to determine whether or not somebody was at a murder scene or committed a crime. So this worked for initial early attempts at identifying people through DNA analysis, but we obviously needed a bit stronger technique to be able to kind of, make some progress on this. RFLP, restriction fragment length polymorphism, is still used today in a very general sense, because this is a very quick way to verify that the DNA molecule you're using is in fact the right DNA. So basically what we saw in the last couple slides with cartoons, this is kind of giving you a real world image here. So here's that agarose gel. This is what the DNA molecule looks like when you don't cut it. So if you just extract some DNA from some hair that you got from a hairbrush sample in your crime lab, and you don't cut the DNA and you put it under an electrical field and you run it, you get a big band. We refer to this as a band right up here at the top. This is your uncut DNA. And then if you cut it, it's going to cut, for example, depending on what enzyme you use, it might cut into 2 pieces. A piece that runs down here at 205 and a piece that runs at 275, and runs at 75, and when you add 205 plus 75, you get 280, which should equal up to your uncut DNA. So in my lab, we will do this a lot when we're trying to confirm that the viral or bacterial DNA sample that we're working with is in fact the right sample. Viruses and bacteria in particular have been heavily mapped over the last 40 years for how they respond when they're cut with certain enzymes. And so this has been widely published. And so you can take DNA from anthrax, from the Bacillus anthracis, the bacteria that causes anthrax disease, and cut it with 3 different enzymes, and you're going to get 3 different bands that should be at a specific spot on the gel. And then you can cut your unknown sample that you think might be anthrax DNA and compare the 2 and say, okay, this is supposed to be anthrax DNA. It came from Dr. Thomas's freezer. I know this is anthrax DNA because we got it from a collaborator. They match up. So for right now, I'm going to assume that this is anthrax DNA. So in my lab, we use this now and this approach for very basic DNA analysis, just to kind of confirm that the sample is what we think it is. But in terms of using this from a criminal perspective, this isn't really used in court anymore, because we can sequence DNA now very, very easily and know exactly what the DNA sequence is. And that's going to be a lot stronger from a criminal conviction perspective in terms of upholding a conviction than it would be from you going into court against Johnny Cochran, the guy that defended O.J. Simpson. And because this happened in the O.J. case, and saying, hey, this DNA that we cut gives us these 2 bands. And when we cut OJ's DNA, we got the same 2 bands. Therefore, this must be OJ's DNA. And then they started asking the question, well, what about the other 6 billion people in the world? If I found one of them and cut their DNA, would they be the murderer? Well, yeah. So this kind of, defeats the purpose from a forensic perspective to be able to use this technique. But this is a technique that is still used in a lot of research labs, because this is, again, something that gives you a very quick answer as to whether or not you think the sample you're working with is what it's supposed to be. Any questions on the RFLP? Okay, and this just kind of gives you some other examples of how this works. Again, you've got these restriction sites indicated with these little dark circles, and you can cut your DNA with enzymes. And these enzymes are very specific. I don't think I mentioned this before, but when you cut DNA with enzymes, the enzymes will only cut in one place, the place they're supposed to cut. Enzymes recognize a very unique codon sequence. Remember those codons we talked about, those A's and T's and C's and G's? Enzymes look at those codons in groups of 6. They look at 6 of those letters, and if they see a unique arrangement of those letters that fits them, then they will cut only at that site. That's why we call them restriction enzymes, because the enzyme is restricted to only cutting in an area where it's supposed to cut. So these enzymes are very specific. So in some respects, you can use RFLPs to generate good fragments of DNA because the enzymes are only going to cut at a very specific place. But again, from a criminal case perspective, when you start to throw in tens of thousands, hundreds of thousands, millions, or billions of people, the chances are always going to be pretty good that you're going to come across more than 2 or 3 people in the world that would show the same banding pattern when their DNA was cut with an enzyme. So we kind of got away from using this technique, even though it does have some utility. Like I said, from a research perspective, it's useful to use enzymes because you can get specific sizes of DNA molecules to drop out. And if you already know what the size of your DNA molecule should be, then this gives you an easy way to confirm. And like I said, from my perspective, when we do research in my lab, we maintain a lot of our DNA as frozen stocks. Well, I try to be very good, my students try to be very good at making sure that whatever is written on the tube is accurate. But sometimes people's writing sucks. You can't read it. And if it's been sitting in a freezer for 10 years, the ice may have rubbed off some of the marker. And you're like, does that say DNA or RNA? Does that say TR or PR? I can't tell. Is this Zika DNA or is this anthrax DNA? One of the things we always do when we pull out a DNA sample from the freezer, even if we know what it's going to be, is we'll screen it with an RFLP analysis just to make sure that we're at least starting with what we think is the right sample. So RFLP can be very convenient if you've got all the materials on hand. If you don't, though, it can be expensive to order. It is time consuming. It takes about 2 days to get this gel done. It takes about 4 hours to pour and then let the gel set. Again, this is basically scientific Jell-O. So you pour this when it's really hot, and just like Jell-O, you got to put it in the fridge for a little while and let it solidify. So it takes about a day to get the gel set up. It'll take you another day to get your DNA cut. And then after you cut the DNA, you have to wash it to get all the enzymes and all of the other chemicals that you use to cut the DNA off the DNA molecule. So doing this RFLP can take about 2 days to get a result. So it can be somewhat time intensive and it can be labor intensive. If you make a mistake and you add in the wrong enzyme, you got to start all over. If you forgot to add the enzyme, I've done all these mistakes, you got to start all over. If you go through the whole thing and damn it, I grabbed the wrong DNA tube, which I've done, you start all over. So it can be very labor intensive and it can drive you crazy if you're trying to get this done quickly. You can also really only look at one area at a time. So it really restricts the focus that you can have in terms of looking at the DNA molecule. You have to have a pretty good chunk of DNA. When you cut the DNA and you clean it and you cut it and you clean it and you run it on a gel, every time you do one of these things, you lose some DNA. It's like walking down the street with a small hole in your pocket and you've got 100 quarters in your pocket. And every time you take a step, a quarter falls out. By the time you get to HEB, that $100 in quarters is now $50. So do you still have enough to buy something at HEB? Yeah, but maybe not what you wanted to buy. So doing DNA work, regardless of whether you're sequencing it or you're cutting it and analyzing it on a gel, every time you handle manipulate the DNA, you're going to lose some over time. And oftentimes, particularly from a crime scene, maybe the only amount of blood you got was a small drop of blood from a wall or that's on a piece of carpet. And you've got to get the blood off the carpet. So you're going to lose DNA just from scraping the blood off the carpet. Then you got to chemically treat the carpet fiber back in the lab to make it dissolve so you can get the more DNA. So if you're talking about something like a biological fluid like urine or semen, these things do not, depending on what environment they're in, they don't persist for very long. If you're talking about blood or semen or urine, on a floor down here in the valley, maybe the DNA would be good for a couple days at most, depending on whether or not it's inside or outside. There's a lot of factors that go into the integrity of the DNA sample. And if you need a large chunk of DNA to do the RFLP, you may not be able to solve the case. So it can be very limiting in that perspective. And you need to have a large amount of sample, and the sample's got to be relatively recent because it tends to break down. And again, from a legal perspective, and we've already seen this play out in real time, these were some of the arguments that were used against the DNA evidence in the O.J. Simpson case. They were able to attack the integrity of the DNA because the cops collected DNA on Friday and then left it in their car all weekend in July in the trunk in California, where it was 100 degrees. So the DNA broke down and degraded. Some of the DNA, they transferred it back to the station, and then it sat in the kitchen in the station for a month. No AC. So the DNA sample degraded. These are all things from a legal perspective that a good attorney will go after if you're using DNA evidence. And so primarily this is why we got away from doing a lot of RFLP work from a forensic perspective. So there's some other things that they try to do to enhance this, but by and large, RFLPs aren't really done a lot for DNA sequencing at all very much anymore. We do use it from some other small things like trying to identify, genetic mutations between children and parents. This is a very good initial screening tool that can be done. So, for example, if there are certain genetic diseases, cystic fibrosis, Huntington's disease, where we know that the gene is coming from either the mother or the father, and we can analyze their DNA by cutting it with enzymes and then running it on a gel to see, hey, does the mother that has this fragment that we know is related to this disease, do any of the children have this same fragment? And if they do, then they may have this disease as well. So it is used in many places still as an initial screening tool. And one thing I think it's worth keeping in mind, I haven't mentioned, is that we're talking about this from the perspective of America. We are a highly developed nation. We have a lot of this technology easily available. We sequence DNA here all the time down here at the UTRGV Brownsville campus. We can sequence the genome in a day. A lot of places don't have this technology. If you go to sub-Saharan Africa, if you go to certain places anywhere around the world, even highly developed countries to some extent, they may not have this technology available. And so DNA sequencing, that is. And so they may be relying on using some of the technology that we kind of refer to as maybe outdated or not as good as what the current stuff is. They're still using this in a lot of areas. So you may not see a lot of this happening in your experiences and opportunities that you see for work here in America, but in other places around the world, they still use a lot of these techniques because they are fairly reliable within a certain context. Just some other examples that I wanted to include in here for y'all as you take this back to communicate this information to your students about DNA fingerprinting. But again, it comes down to, even if you've just got a piece of the DNA, from our perspective today, you really need to have the whole genome so you can cut and get as many of these bands to show up as possible if you're doing a comparative analysis to try to identify paternity or to link somebody to a murder case. One of the advances in DNA technology that's helped to drive a lot of the benefits that we enjoy today is polymerase chain reaction, or PCR. And what you're seeing here in this image is a old PCR machine. But PCR again stands for polymerase chain reaction. This is an experimental technique that we use to amplify DNA. So let's say you show up at a crime scene and you've been able to extract some blood from the wall. You found a blood spatter. You cut off the piece of wall that has the blood spatter. You get back to the lab. You spend a day or so shaving the sheetrock as much as you can to get that blood drop away from the sheetrock. And then you're going to use chemicals to try to break down the paper that the blood is attached to so you can kind of release the blood droplet. and then try to extract the DNA from the blood. And let's say that you get some DNA from the blood, but it's not a lot of DNA, and you need to amplify. You'd love to be able to amplify to make more of the DNA. Traditionally, we didn't have this ability to do so, but in the late 80s, PCR was developed. And again, in the mid to late 90s, PCR really became a very popular, very commercialized technique that really made it easy for a lot of people to do. And what PCR will do is it will allow you to amplify the DNA that you have. So if you've gotten some unknown DNA, you can add some chemicals to that DNA solution, and then you can put it in a thermocycler, a PCR machine, and it will amplify the amount of DNA that you have in your sample, thereby giving you the opportunity to maybe do some testing that you couldn't do initially because you recovered such a small piece of sample. One of the main areas that we see this used today, not just with forensic DNA for crime scenes, but is in when we find bones from human beings from 150,000 years ago or something like that. Has everyone heard of the, you probably heard of the direwolf pups? Has everyone heard of this? Does everyone, dire wolves? This is a breed of wolf that died out about 15,000 years ago at the end of the last ice age. And we have found, just like we found with woolly mammoths, we found some of these animals frozen in ice. And scientists were able to go, they found a direwolf cub that was almost completely preserved in the permafrost. And they were able to extract DNA from that direwolf cub. And then they were able to amplify that DNA, and they made enough of it to where they could take that DNA, and just like we did with Dolly the sheep 30 years ago, they cloned that direwolf DNA into an existing direwolf, into some wolf eggs, fertilized wolf eggs, and were able to regenerate direwolves. I don't have any of those images up here, but they announced this about 2 months ago. It's kind of cool. You can actually go online and see they got 2 puppies that were born from this cloning procedure, and they were actually about a year old now, and they're huge. Dire wolves were about 2 or 3 times the size of a normal wolf, and they've got these dire wolves roaming around. Their plan is to make about 12 of them, so they can kind of study how they interact and what they look like and what they sound like and that kind of stuff. But this came from their ability to take that small bit of DNA that they were able to get from this direwolf body from 15,000 years ago and amplify it using PCR to make enough DNA to where they could use it to clone into a fertilized wolf egg. So PCR has really been a big factor in allowing us to do the kind of work that we do in DNA work in DNA amplification today, because we can take basically the smallest piece of DNA that we've been able to isolate and use it to build out and amplify, and then make a bigger piece of DNA that we can study. And that bigger piece of DNA is going to be very, very almost 100% identical to the original piece. And so this lets us take unknown pieces of DNA that we may only have a small fragment of and make a lot of it so we can study it, we can manipulate it, we can see, and what we want to do with it. So this is just kind of some information about when the forensic community does PCR, they focus on very specific regions that are found within all humans to amplify, because from a forensic perspective, if you've got an unknown piece of DNA, you may only get one chance to amplify it. And you want to make sure that you put everything in there in the proper concentration to get the amplification. And you need to have a target region to amplify. If you're going to try to make more DNA, you can't just mix everything in there like you bake a cake and say, okay, I put in more flour, I put in more sugar, now make more DNA. You have to tell the molecules where to go on the DNA molecule to be able to start making more DNA. And so from a forensic perspective, the forensic community in the United States and pretty much globally has settled on what they refer to as a tetranucleotide repeat, which is AATG, and this AATG is repeated over and over and over and over and over and over and over in certain stretches of DNA for everybody. for every human sample that's been analyzed. And so if you're doing this forensically, you would use this AATG as kind of your starting area because there's a very good chance that the tiny blood spot that you got that had a little bit of DNA probably has this AATG repeat in there that you can use as a starting point to make new DNA. These areas in the DNA molecule where you see a repetition of the bases, AATG, over and over. And these are literally right next to each other. This says 7 repeats right here. So 1, 2, 3, 4, 5, 6, 7. That means that this is AATG, 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 and so on, and so on, and so on, and so on. And so we call these regions in DNA where you have these constant repetitions of 3 or 4 bases, short tandem repeats, because they're short, they're in tandem, they're next to each other, and they're repeated over and over and over and over. And this AATG tetranucleotide repeat is what the forensic community basically uses anytime that they're going to try to amplify an unknown piece of DNA. So I talked a little bit about this already. What is PCR? It's basically a way that we can amplify DNA. Even if you have just a tiny amount of DNA, the chances are very good that you'd be able to amplify enough DNA within that mixture to be able to get some kind of a reaction, to be able to do some kind of work on. And basically, the process of making DNA is a 3-step process. We have to first heat the strand up, and we're going to look at a little video here that'll kind of show this here in just a moment. And I've included these links here in the slides. I'm hoping that these links will work for y'all. If there's any issues with these not working, what I'll probably do, and I'll go over this when we get to the end of the course, when we talk about some of the resources that are available, I'll make sure to include functioning links for these resources on the Center for Genomic Resources Research's webpage that y'all will all be given access to when we finish the course. So you can go in and use these materials in case you need to use them for educational purposes on your own. So let me see if this animation will even work. And it's not. It's going to. Okay. I'll have to get this link up and functioning. But I will have these links on here so y'all can go and look to these. These things link back to this links back to Cold Springs Harbor. Let's see where this one takes us to. Okay, so this one actually does work up here. So I won't bore y'all to death with watching this video, but this is great for you to use for students if you've got them in class. You'll have a 3-minute, 27-second chance to go to the bathroom while they watch a PCR video. But basically, we'll go through this here. PCR basically occurs in 3 phases. Step 1 is you have to heat the DNA molecule, because again, remember, the DNA molecule is a double helix. It's these 2 strands that are wrapped together, that are held together. The bases, the A's and the T's and the C's and the G's, are held together by these hydrogen bonds. And the reason for the hydrogen bonding is that it's strong enough to hold the molecule together, but it's not so strong that it can't be broken. What's one way that you can break hydrogen bonds? Through heat. So the molecule is heated up in the thermocycler machine, and this causes the 2 strands to denature and to separate. So your 2 strands that are wrapped together get heated and they come apart. And once they come apart, you then have chemicals that you've added in there, enzymes that are used to replicate DNA. They will go in and they will start to bind to specific areas on the DNA molecule, and then they will start to replicate and make new strands of DNA. So we first have the denaturing, where the strand is heated up and comes apart. And then we have the amplification stage, where the new strands, we have our strands come apart into 2 different strands. And now these 2 strands are going to be amplified. And then once the growing phase, if you will, the amplification part of the DNA process is complete, then they're going to come back together. They're going to hybridize, anneal. They're going to come back together. And now you're going to have 2 new strands of DNA that are going to be identical to each other. And this process occurs over and over and over. This is how DNA is copied in the body. We're just kind of doing it artificially in a PCR machine to be able to make more DNA. So step one, the molecule gets heated up. It gets heated up to, I apologize for the spelling error here, minutes, I guess this is the French version. We heat the DNA molecule up to 94 degrees Celsius, so pretty hot. 100 degrees Celsius is getting pretty toasty up there, right? That's boiling. So we heat it up to 94 degrees Celsius, and this causes the double helix, the bonds, hydrogen bonds between the A's and the T's and the C's and the G's gets broken. And so these two strands can unwind and come apart. So now we have a strand over here and a strand over here. And then we have molecules that we've added into our PCR mix that now will come in and they will recognize this DNA strand over here at this end and this end over here. And it will start to using this as an instruction template, just like we saw in the beginning of the talk, start to make a new DNA molecule that's complementary to this strand. So remember, if this is an A and a T, then this is going to be, this is an A recognizes T. So that means if this is an A, it's going to make a new T and then a new A. So enzymes will come on and sit on these two separate strands and read these bases as an instruction manual. And if this says an A, it's going to add a T. If this is a G, it's going to add a C. And it will make a brand new strand using this as instructions. Oops. And it will also make a brand new strand using this as instructions. So the end result is going to be a new strand that's part new, right? This is your new stuff, but this is the old strand. This is the old piece that was used as the instruction template. See that up here? So now the initial strand that was used when you denatured, that was used to make the new strand, is now going to be mixed with the new to form a new molecule of DNA. And the same thing happens down here. So when you make a new molecule of DNA, one of the strands is going to be a newly made strand. One of the strands is going to be an old strand, the instruction strand. For this reason, we have a new strand and an old strand that comes together to make a new DNA molecule. We refer to this as being semi-conservative. And this is called the semi-conservative model of DNA replication. All that means is that when you're making a new strand of DNA, one of those strands is going to be the new hotness. The other one's going to be the old busted funky. So you're conserving one of the strands to make a new strand. Since we're conserving one, we call that semi. So the model is referred to as the semi-conservative model of DNA replication. So, and the same thing happens in the body. We're talking about this happening in a machine, but the same process is occurring biologically. So the strand is denatured in our PCR machine, it's heated up, and then we have the annealing process where all of our enzymes come in and they start making new DNA strands using the old strand as templates. They make a chunk of DNA, and then they fall off, and then they get back on, and they make another chunk, and then they fall off and get back on and make another chunk, and then all these little pieces get reassembled back together properly to form our new strands down here at the bottom. And then the new strand is going to be connected to the old strand. We're now in a process called the extension, where we're just making new bases. We're adding to where the new strand is going to be just as long as the old strand on both sides. And if you look from a time perspective, it takes a minute to denature. It takes 45 seconds to start to make the new copies. And then you let it run for about 2 minutes to finish making the new strand. This is referred to as one cycle. Denature, anneal, extend. This is one cycle. In order to make a complete DNA molecule, it needs to run about 30 to 40 cycles, primarily in step 3, which is the extension part. So if you look at this from a time perspective, if you're going to run this for 30 cycles, you're going to spend 1 minute times 30 cycles at 94 degrees C. You're going to spend 45 seconds times 30 cycles at the annealing, and then 2 minutes for 30 cycles, the extension. So what you would do is then say, okay, This part is 30 minutes. This part, 45 seconds, is going to be what, about 20 minutes, 25 minutes. And this is going to be 2 minutes for 30 cycles, going to be 60 minutes. So this is going to take altogether maybe about 4 hours to run. So in about 4 hours, you can take an unknown piece of DNA and amplify it into millions, if not billions, of copies of your full length DNA molecule. And this right here, the ability to take DNA and stick it in a tube and throw it in the machine and push a button and walk away and come back in 4 hours, is what's really driven the large benefits that we enjoy now with DNA sequencing and drug discovery and being able to identify crime scene victims, being able to identify dead people, being able to identify paternity, who are children related to and that kind of stuff. All of this has come from this process right here of PCR. If we could look back in 100 years, assuming we're all still around and we're not taken over by Skynet at this point, and try to identify a seminal moment in medical history or biomedical research history that really revolutionized it, this is going to be one of these moments where PCR was developed and created. This has been a huge leap forward in giving us the ability to basically copy and make DNA molecules from anything. And so when you see stuff happening like in the movies, like in Jurassic Park, where they go and they find an insect stuck in amber and they suck DNA out of it and they make more DNA, which they have done, this is how it's being done. And this process is being done everywhere around the world. This went from being something in the 90s that you did with a machine that was half the size of this table, just to now being something that you can do with a machine that's just a little bit bigger than your smartphone. And we actually now have the technology where this can be done in a portable environment. So I've done work in the jungles where we've gone and collected insects from the upper canopy of trees, and then we can come down, climb down the tree, mash up the insects, put the insects in a PCR machine, extract the DNA. This is all in the jungle. Run the sample in a thermal cycler, amplify the DNA, and then put it on ice, and then save it until we get back to the main lab. So the ability for scientists and researchers to be able to do this has really revolutionized how we approach science, particularly as it relates to DNA. So this is going to be probably one of the most important concepts and one of the most important discoveries that we've made in the 20th century, moving forward for science. And this is, again, just kind of giving you another idea of how this can go. So you can start from a single strand and get 68 billion copies in about 4 hours. Very, very powerful. Very, very powerful technique. This is just kind of some graphics, some small animations to kind of give you all some ideas for how this part is functioning. All of these different colored dots you're seeing in here is meant to represent the bases, the A's, the T's, the C's, and the G's. And what you're basically seeing here is that you get the denaturing part where the strand is unwound. You then have, here's the denaturing, it unwinds. You then have the annealing where you're getting your new strand formation starting to begin. Now you have the extension where the new strands are growing, and the end result is going to be your two new strands formed for your two new DNA molecules that were made from one. And again, one DNA molecule is going to have the old strand and the new strand. So a nice little animation here. This might be useful for y'all kind of moving forward. This just kind of, again, looks at some of the stuff that we've determined from the RFLPs. We've kind of gone through this a little bit more, so I won't beat this to death again. Again, this is something that can be useful in some environments from a forensic perspective to identify a suspect, but you're not going to get the hard yes or no that you would get from doing DNA sequencing. DNA fingerprinting has also been used in paternity. And again, we've kind of talked about this a little bit more. For paternity, it's a little bit more useful. This graphic is kind of meant to show you if you get this banding pattern from mom's DNA. So you take mom's DNA and it's represented with the egg here. Dad's going to be recognized with the sperm cell. And you were to cut it with restriction enzymes, mom's going to give you a very specific DNA banding pattern. and you cut dad's, and dad's gives you a different pattern. And then you look at the 4 kids and you can say, okay, child 1 has got this band that correlates with mom. This one correlates with dad. Oh, look, this correlates with dad. You got a match out of these 4. So you could probably say that this child is the result of these 2 offspring. And then you look at D2 and you say, okay, this kind of matches dad, but it doesn't have this band from mom, but it has this band from mom. So, okay, this child matches up to the mother. So this is probably the maternal descendant from this parent, but we're not seeing this from dad, right? We're not seeing a band here. This band here is slightly different size. This is a slightly different size. We are kind of cutting hairs. And again, this is why we got away from using RFLPs. But as Maury would say, you are not the father, more than likely, of daughter 2 right here. We look at son 1, and again, you can kind of line up the banding patterns over here. And you can do the same thing with son 2 and say, again, this is probably not the father, as indicated by the green egg and the red sperm over here. So again, this is a very simplified version, but RFLPs, as I mentioned, they have some utility, but if you're going to go on the Maury show, you want more confidence than this. I mean, at the end of the day, again, you're asking someone to make a judgment as to whether or not this is this band is higher than this, or this band is lower than this. I mean, you're looking at size differences that can be difficult to gauge, and you can artificially mess with the sizes by running the sample for longer than it's supposed to be. If you run the DNA sample under electrical field, it'll continue moving even when it's gotten to its size. So if you run it for too long, you can run the fragment longer than it should be, and you can alter the results. And again, this is one of the reasons why we got away from using RFLPs. But this is still a technique that's used in a lot of places around the world because DNA sequencing is an expensive operation. It needs very specific requirements in terms of power, computing power, the ability to process and store the data. And DNA sequencing machines by themselves are not cheap. They run about $150,000 to $250,000. And you need to spend about $30,000 a year just to maintain the machine. So if you're talking about a lab in Kathmandu or somewhere in the hinterlands of Pakistan, they may not have the ability and the resources to buy DNA sequencing, but they can get enzymes and get a PCR machine and do RFLP analysis. So you will see some of this still used around the world, but it's not going to be as used as it would be for DNA sequencing. So just kind of another little example here showing you the DNA fingerprinting technique, and it's being used up here where you can kind of walk through this and see this is what you get from sperm cells. This is what you get from a known sample. This was the boyfriend. These are some control samples. This is the victim sample from the 2 suspects, and they show you over here that they were able to use this. And as I said, you can get some data from this, but at least in the American legal system, we've seen that a good attorney can rip this to shreds. And so now a lot of our DNA analysis and identification is done exclusively with DNA sequencing, because when we sequence and we actually read the specific DNA sequence, we can say with a very high degree of certainty, greater than 99%, that, hey, this is the culprit, this is the guilty party. Capillary electrophoresis is another DNA technique that you'll see used, particularly in forensic settings, which is basically very similar to what we talked about with the RFLPs. The difference here is that a laser is used to basically bounce off of the DNA molecule, and the laser beam can track the DNA bands as they separate. And what this does is this lets you analyze very, very small fragments of DNA. When we're looking at these DNA fragments here, and I apologize for not mentioning this sooner, it is indicated a little bit here, but I should have probably gone over this a little bit more. These are DNA fragments that are probably several hundred to several thousand base pairs in length. So we're talking about, this could be 5,000 A's and T's and C's and G's. These down here are in the hundreds. This may be 400 or 200. You'll get DNA fragments that could be 10, 8, 5. We generally don't look at those because they're so small and tiny that they run off the gel. Again, you have to keep in mind that you're doing this is done on a piece of scientific jello that has an electrical field. The DNA samples are all put in here at the top, and the electrical current forces the DNA molecule to move down the gel. The bigger the molecule, the slower it's going to go. The smaller the molecule, the faster it's going to go. And if you're trying to track down a fragment that's less than 100 base pairs, that thing moves like Superman. It'll run off the gel so fast that you may miss it. I can't tell you how many times as a graduate student, I would run a gel trying to capture a 50 base pair fragment. I would go to the bathroom to pee and come back in 5 minutes and the damn thing is ran off the gel. And once it runs off the gel, it's gone. You have to start all over. I mean, those fragments move quick. And even if you can catch them, there's only 4 or 5 bases there. That doesn't, if it's just ATCGG, that may not tell you a whole lot. So a technique was developed to be able to look at those small fragments and use a laser to scatter light off of the small fragments, which is then captured by a machine that can tell you what those base pairs are. And this is referred to as capillary electrophoresis. So it's kind of a combination of the RFLP technique, but you're shooting those fragments with a laser. The big advantage is that it can detect bases that are very, very small, very, very small. The bad part is that the bases are very, very small. And if you're only getting ATCG out of a sequence that's 3 million letters long, you're going to find ATCG probably repeated tens of thousands, if not hundreds of thousands of times in that DNA molecule, which may not be very useful for you if you're trying to tie it back to a criminal suspect. This just kind of gives you an idea. When you scatter it with a laser, you're going to get, and this is indicated by the different colors, you're going to get 4 different wavelengths. One of these is going to be for the A's, one will be for T, one will be for C, and one will be for G. How do you know? Because they use a different colored dye. When the laser hits the DNA molecule, depending on what the base is, it gives off a different color that a computer can capture and then interpret. So if the laser hits the molecule and it comes back as blue, then those are A's. It hits it, comes back as green, then those are T's. The blacks are C's, the reds are G's. And so you can then use this data to say, okay, we got an A, and then a T, and then, oh, then it looks like we had a G, and then a T, or then a C, and you can use this to kind of reconstruct what the DNA sequence was. Again, this is stuff that we did in the beginning of time, so to speak, in the 90s when we first started to do this. This is not easy stuff to interpret. It's very much open to the experience of the person that's reading it. And from a legal perspective, if you're an attorney, you would love to see this kind of stuff in a court setting, because it's very easy to attack this as not being very specific. And this is, again, why you don't see this used very much in the United States anymore. Almost exclusively, you will not see this used in criminal cases in the United States when they're trying to identify DNA back to a specific subject, because a good attorney can rip this apart. But this is a technique that still gets some usage within some places. And these are just some follow-up slides to kind of show you how they would put this together and basically build the sequence back from using these small fragments and the laser scattering properties to kind of tell you what these are. So, and as I mentioned, this can be useful to some extent, but it is going to have some limitations, and it's going to really simplify your ability to interpret what the DNA sample means. And so it can be useful. For example, I think the example that's given here is if you have maybe you have a criminal complaint with a female victim, and she is alleging that she was maybe sexually assaulted by multiple males, and you want to see who exactly was involved in that. You've got those males grouped together as suspects. You can run an initial profile like this very cheaply and quickly to eliminate 2 or 3 of the subjects. For a lot of places, and I know I said for here, we can sequence DNA very cheaply and very quickly. A lot of places don't have that ability. So even in America, they will still use this technique initially to take a group and maybe reduce it from 10 suspects down to 3. And then they'll take those 3 suspects and run a full DNA sequencing analysis on them. So this does have some utility and it does get used in a very, very basic way. But DNA sequencing is still the best way that we go. I think the last one that we'll look at getting. Sorry about that, Wanda. We'll be looking at forensic DNA and then we'll pick up the rest of this sequence probably tomorrow. We'll see where we're at here. I apologize for skipping around here. Yeah. Okay, so we're close to being finished up. So the last application I want to talk about forensically for DNA sequencing, which is something that is actually kind of stepping into the modern realm. And this has a lot of applications outside of the criminal perspective, is mitochondrial DNA sequencing. I apologize if I'm getting anybody a seizure here. I'm trying to get back to the slide here real quick. Is analyzing mitochondrial DNA. So for the just for the purposes of clarity, to make sure we're starting from the same space, everybody knows the body's composed of billions of cells. Within each of those cells are specific structures. One of the structures that's found within virtually every nucleated cell within the body is the mitochondria. This is often referred to as the powerhouse of the cell. This is the organelle, the structure within the cell that's used for making energy. It's used for cellular respiration. It's going to help make ATP. Mitochondria is found virtually, like I said, in the cytoplasm of every cell. The mitochondria can self-replicate. It has its own DNA. And what's unique about mitochondria is that the DNA in the mitochondria you inherit only from your mother. If you think about all the DNA in your body, it is a mix. You get half your DNA. We're saying half for the purposes of discussion here. It could be a little more, a little less, but generally half comes from mommy, half comes from daddy, and that's where babies are made. Very generally. The mitochondrial DNA only comes from your mother. So if we look at mitochondrial DNA, this is looking at the maternal line of your family. So the mitochondrial DNA that you have came from your mother, and if we look at it from your mother, it came from her mother, and so on and so on. And so since mitochondrial DNA is inherited from the maternal side, you can use this when you look at relationships within families and you look at ancestry to see where changes start to occur, because the DNA in the mitochondria is coming from the previous maternal generation, from your mom, from your grandma. So it's very, because the DNA is only coming from your mom, it's the analysis is much more sensitive, it's much more robust, because you don't have to worry about, well, some of this came from dad, some of this came from mom, but it is tougher to do. A little bit more rigorous, it's tougher to get the DNA from the mitochondria, a little bit more time consuming, a little bit more costly, but it is a very, very powerful tool in forensic analysis and just in analyzing DNA in particular. I don't know if y'all have heard of this. This is a hypothesis that's been thrown around a little bit. It kind of gets misspoken here and there, but this is referred to as the Eve hypothesis. And this hypothesis basically states that everybody walking around alive today is descended from a single female that lived somewhere between 80 to about 100,000 years ago. Is this true? It is. Now, does that mean that we are all, that one woman like made all of the people on the planet? Of course not. But we know from analyzing mitochondrial DNA that all of the mitochondrial DNA that currently exists on the planet right now traces back to a very small population of humans that lived somewhere around between 70 to 100,000 years ago. How do we know that? Because we've looked at mitochondrial DNA samples from skeletons that have been recovered that are 200 to 300,000 years old, and they're completely different than the mitochondrial DNA that exists in everybody today. And what they think happened is that about 100,000 years ago, there was a massive volcanic eruption called the Togo event. And this is a volcanic eruption that's never been seen before. It wiped out about 95% of everybody living on the planet at that time. And it caused what's referred to as a genetic bottleneck. If we look at mitochondrial DNA prior to this volcanic eruption happening, there's a lot of variation. And then you have the volcanic eruption occurring, and that variation goes like this, and it gets almost non-existent. And it's like that for about 20,000 years. And then the variation in the mitochondrial DNA starts to spread back out. That bottleneck event that occurred took the human population globally down to about 2,000 people. So sometime about 100,000 years ago, we went from maybe 1 billion people on the planet to about 2,000 people. And everybody walking around today is descended from those 2,000 people. And the mitochondrial DNA comes from the maternal DNA that was present in that population. And it's all related. So what this tells us is that the 2,000 people that survived this volcanic eruption, this calamitous event that repopulated the earth, were all from the same family, basically. They were not, I'm not saying they're all inbred, but they all were genetically related back to a single female. So you could imagine maybe there was a family of maybe a tribe of 100 people, and maybe within that tribe, most of the women, all the women were descended from a single female that maybe had 20 kids, and she had 10 daughters, and she had 10 daughters, and they went out and married different people from the tribes, but they all came back and lived there. And whenever this environmental event occurred, they were together, and their mitochondrial DNA is now what forms the foundation of every mitochondrial DNA sequence that we see alive on the planet. So if you get some time to look it up, it's called the Eve hypothesis. It's a really interesting hypothesis. I don't know if it's accurate or not, but the thing that we do know is that we do know that this bottleneck occurred. Something caused the global population of people to go from about 1 billion to 2 to maybe 10,000 people. And it was like that for about 10,000 years. So pretty interesting. But because of this highly conserved nature of mitochondrial DNA, this is also used in forensic analysis to be able to tie and connect people back to each other. And so a huge database has been generated over the course of the last 20 years that the FBI maintains that's mitochondrial DNA sequences. And this is used to be able to identify DNA samples, particularly if you don't have a good nuclear DNA sample available for testing. So this database is called CODIS, and this is what the FBI uses. So when you sometimes see in the news, hey, they just found the killer, this unsolved murder from 50 years ago has just been solved, and they identified who the killer is, it's because now people are going to sites and companies like 23andMe and getting their DNA analyzed. Well, this information has been shared with the FBI, and the FBI can take an unknown DNA sample, sequence the mitochondria, put it in its database, this CODIS database, that's collecting data from all these companies, and then it can use that to say, hey, we got a hit. And so then they can go to the people that submitted that DNA sample 23andMe and say, hey, did you have an uncle or a brother that maybe was alive at this time, this and that? Oh yeah, we did. We have some of his stuff. He has a box of old uniforms upstairs. He's dead now. And they're able to go back now and sequence the DNA and say, hey, this might have been Jack the Ripper. This might have been the killer. This is actually just used to maybe identify who Jack the Ripper was. They have been able to use this sequencing technique, and they think that they've identified and confirmed that Jack the Ripper was one of the suspects. It was a Hungarian-Polish barber that they had looked at when these killings happened in Whitechapel in the early 1900s in England. And they had maintained some of the knives and some of the bloody clothing from the victims, and DNA sequencing had always showed that some of that DNA that was on there was not related to the victims, because they have descendants of the victims that they got DNA from and they compared it to, And so they just used this technique. I completely forgot about this, but they just used this technique a few months ago. You can find this on YouTube to identify who they think was Jack the Ripper. So this CODA database is used by federal law enforcement, as well as I'm sure Interpol and other law enforcement companies or companies around the world to identify DNA sequencing. And a lot of this is done from the mitochondrial DNA samples. Because the mitochondrial DNA is only inherited from the mother's side, there's very few changes that occur in it over time. And we can use the lack of changes to be able to track back to identify who a given DNA sample comes from using the mitochondrial DNA. Again, it's not as easy to get, but it does have some utility beyond what we see. And something that we'll need to keep in mind, and we'll talk about this in some future talks coming up, is we now have had all these companies that have been created, like 23andMe, like NewGene, that you send in your DNA sample, right? They send you a cup. You can spit in a cup. You can pee in a cup. They'll send you a Q-tip. You can swab your cheek. You can send in the sample to them, and they'll sequence your DNA and then send it back to you. Well, I don't know if y'all have heard or not, 23andMe went out of business. And so now there's a very big, heated discussion over who owns that data. They have done hundreds of millions of DNA sequencing samples. That is a treasure trove of data, not only for drug discovery, but for insurance companies. Insurance companies would love to be able to get a hold of your DNA, because if they can see your DNA and say, hey, Luis, you are prone to having diabetes in your family. We're not going to insure you for diabetes, or we're going to raise your insurance rate because you're more prone to getting diabetes because your great-great-grandmother has diabetes. This is the kind of information that that the public doesn't want to have companies getting a hold of, because they can use this to go after people, to raise your insurance rates, to maybe deny you healthcare coverage, or for some other kind of nefarious purpose. And so this is something we're grappling with right now. We'll talk about this in a presentation we'll do later on the mini course about some of the ethics involved in genetic research. But we now have a lot of people that have sent in DNA to 23andMe. 23andMe is filed for bankruptcy, and so now they're fighting over who owns that DNA. And whoever wins that bidding war, who knows what that information is going to be used for. I mean, it will eventually probably find its way to insurance and healthcare companies, who will eventually use it to probably discriminate against giving people insurance. And so these are all things we're going to have to think about as we revel in the revolution of DNA sequencing and all these great things that we can get out of this, that we're going to have to kind of think about moving forward. This is some information I wanted to include in here. There's some examples of some DNA calculations. I really wanted to throw this in for any of the teachers that are coming in. These would be some great things that I think you could use from assignments for high school students, on how to be able to do some of these examples and some of these basic calculations. So I had thrown this kind of in for the teachers. But this kind of wraps up the discussion as it is. So I wanted to go ahead and let you know that all this material will be made available to y'all once we complete the course. The slide set will be made fully available, and as I said, I'll go ahead and kind of get the links updated as well. We've only got a couple minutes left, and I apologize. I know we've got a few minutes over, but I'd like to give people the opportunity to ask any questions that they might have or anything like that.
Will you be emailing us those slides?
I will either email them, or we have a website for the Center for Genomic Research that we actually have up and running. We recently moved over from the School of Medicine to the College of Sciences. And so because we moved from the School of Medicine to the College of Science, they had to go and redo the website and take off everything that says School of Medicine off of there. So we had the website up, we took it down. They're telling me it's supposed to be back up by the end of the month. So I will for sure share all the slides with y'all when we finish up the course. So you'll have that. And then you will have the ability to go on to the Center for Genomic Research's website. And we have some additional materials that will be stored on there as well. And I will also put all the slides and any supplemental material on those websites, on the website as well. So I guess the long, boring answer is yes.
Okay, thank you.
You bet. Any other questions about stuff?
I have another question. So I'm in the Brownsville campus and I was supposed to go in person, but I couldn't find the place. Is it the building behind the life and health science?
We are connected to the Luis Colon, to the Salone Cassia building. So I don't know if you know, there's a brand new building for human genetics that's been built on the campus. We're on the backside, next to Jackson and the bridge. That
Oh.
backside, do you know where the police station is?
Yes.
So if you are driving, and the police station is on your left-hand side and you're driving to the campus where that big circle is, turnaround is, if you park in that parking lot right there, the building is right there.
Okay, so it was a building that not too long ago was like covered.
Yes, it was under construction, yes.
Okay,
So
okay.
if you, and I assume, Raul, or Luis, you're Raul. Raul, you're gonna be here tomorrow too, right? So Raul and I will both be here, Karina. Should be here probably at 10:45 or so. So I'll keep an eye out for you in the front, and so you can just come up and sit here in the room with us.
Okay, that's perfect. For now, I'm in the like the library, and I was like, I just couldn't find the building.
Yeah, that's okay. And for those of y'all that are in the McAllen Edinburgh area, I know it won't be much help for y'all, but we will be running another series of the course in August. And then from this point moving forward, every March and August, we only did it May for this year because we had some delays getting started for the grant because of the presidential election. All federal grants had to go through a review process to make sure we weren't spending the money on stupid stuff. So we were delayed in getting the grant going initially. So only for this year, we're gonna do it in May and August, but moving forward for the next 4 years, we'll do this in March and in August. We can do it during the week of spring break. I know it's kind of a shitty way to spend spring break, but at least people are out of school and hopefully out of the classroom if they're a high school teacher. And then we'll also do this in August as well, probably the first or second week of August. So again, people have the ability to attend when they may not be in class or might not have other commitments. So next session will be live in the Edinburgh campus. But we'll look for you here tomorrow, Karina, so you can come up and sit with us here in person. And yes, Miss Wanda put in the chat, anyone who wants to come in person, if you can't find it, she's put her phone number in there. It's 956-882-7541. Her name is Wanda Wiley. She's a fantastic administrator that we have here. She's only gonna be here for a few more months. She's retiring and escaping the madness. So we'll have her here to help us for a few more months. But if you do get lost and can't find it, look for Wanda. Look for Wanda's number, and then I'll be out in front of the building tomorrow and I'll keep an eye peeled for you.
Okay, thank you so much.
You're welcome. Any other questions or comments before we wrap up for the day?
I had a quick question. So you mentioned that 23andMe went bankrupt, correct? Do you know who
Yes.
paying for the store all the database information?
So right now, because they were a publicly traded company, they had investors, and so there's when a company goes into bankruptcy, the courts generally take it over and they put them in what's called receivership, and they take whatever money they have aside and put that towards maintaining all of their materials, whether it's equipment. If you were a construction company, they make sure that the trucks are all being stored and that kind of stuff. So the courts have put them in receivership, and so they've had to put money aside to maintain the samples, and so they're maintaining the samples. But what's really the value is really not in the biological samples, it's in the data that they got from it. And so that's all being maintained as well. And the question is just right now, it's just a big legal fight over who owns that data. Do the creditors, the investors that put into 23andMe own that data? Does the company own that data? If it's the company or the investors, under US bankrupt and most global bankruptcy laws, if you go bankrupt, you got to pay everybody back that you owe money to, which means you got to sell all your crap. And that includes that data. And that's where the trouble's coming in, because that data could be worth like $1 trillion. I mean, that data insurance companies would love to get their hands on, because even though they're not supposed to use it to discriminate against us, and I'm not trying to beat up on the insurance companies, but we all probably have insurance. We pay insurance. I know insurance down here, every time I open my eyes, my car insurance has gone up $50 for something. If they can find a reason, I've been paying higher insurance in Texas for 40 years because they keep telling me that half the people driving around in Texas don't have insurance. But it's been like that for 40 years. I mean, either fix the problem or don't let those people drive, but their response is just, well, we're gonna let them drive, but just make you pay more money. So what? But because of that approach, they're gonna want to probably sell that data and get as much money for it as they can. I mean, the guy that owns that company would probably like to be able to retire with something, I'm assuming. So they have not decided what they're gonna do with it. It's a huge battle, and we will discuss in great detail, not great detail, but we'll discuss about it in some detail in one of the lectures we'll do later on this week about what's gonna happen with that, because it's a very big problem that a lot of people in the genetics field are worried about, because whatever they decide to do with them is going to have far-reaching implications for everybody else. We do DNA sequencing for cancer patients. That's a great way to identify when someone's on chemotherapy or taking some kind of chemo drug, if the drug is causing mutations in their cancer cells, to sequence their DNA. Well, that is data that's being generated. Are we going to be forced to give that data over at some point? Could the state come in, because we are a state entity, and say, well, you have to give that data to Blue Cross Blue Shield of Texas so they can decide whether or not people get coverage or not? There's a lot of ethical questions that come into play when you start to look at this. So it's going to be really interesting to see what they decide with 23andMe. And it's funny because my mom, my sisters, virtually everybody in my family went and did that. And I actually got the kit from my mom for Christmas for like 3 years ago, and I never filled it in. I just threw it in my closet and forgot about it, because they wanted you to put in like this much spit. And I was like, I'm not gonna, sit there for 30 minutes and spit in a cup. F that. I looked at what my mom's DNA sequence was and my sister's and that kind of stuff, and kind of said, okay, this is what I'm looking at. Now I'm kind of thankful that I didn't do it, because who knows who's gonna get that data? Probably some dude in China. So they may have it already, right? So, but it'll be very interesting to think about. We'll see.
Yeah, that makes
Sorry.
Oh, I was gonna say that makes me wonder how often like this information is even updated with like these insurance and health companies, and like we were talking about the CODIS from the FBI. I wonder how often this information is updated.
Yeah, that's a great point. And I would assume that they probably have some kind of federal policy. I'm sure that they do, that says CODIS will be updated on this basis or that basis. But just look at what we've seen coming out of Doge for the last 5 months. I mean, who would have imagined that the government's still using paper records? I mean, everyone saw that, right? That there's a mine in Utah that all the government records are stored on paper, and the paper records are stored in this mine that's like half a mile deep in the earth. And because they had asked the question, how come only 10,000 people can retire from the government every month? And they said, well, how come? There's 2 million people that work for the government. How come only 10,000 people? Because somebody has to go down the elevator, walk back in the cavern, get the paper record, walk it back to the elevator, ride the elevator back up, and then use those paper records to help the person retire. There's only enough space on the elevator for 10,000 records a month to go up. So the Social Security system was using computer systems from the 1980s. I mean, would anyone like to? I was there in the 80s when computers were invented. I can tell you, I would not want to use a computer from the 80s to do anything, not even sharpen a pencil. But this is what we're using to send out IRS checks. And so you could probably imagine, in my mind, you always think of the government should have the best of everything, right? They probably are using great supercomputers. No, they're using computers that are like from the 80s and stuff. And so if that's the case, then that database maybe is never updated, or maybe it only gets updated once every 5 years because the computers won't allow it to be easily updated. You would assume, like a lot of us do, that, hey, if I call and log into my Apple account and I delete photos off my iCloud, they're gone instantly, right? I can pull up my phone and those photos are gone. If I make changes online to my academic record at UTRGV, those changes go into effect immediately. So we all kind of have the assumption that the government and state agencies kind of function the same way. And now we're seeing that, particularly with the federal government, they haven't spent the money on buying new computers. They spent it on doing other silly things instead. And so maybe the database can't be updated because it's really tough to do. Maybe they can't even use a flash drive. Maybe they have to use a floppy disk or something from the 80s to update it. Y'all probably don't remember, but before floppy disks, we used cassette tapes. to load data into computers. I still have video games for a computer that my parents bought me in 1981 that's on a cassette tape. You can't play it on a cassette player, but you can put it on a cassette player that hooks to your computer and it plays this video game called Telengard. They came on cassette tapes. That's the technology that the government is using for some of its computing systems. So,
Is that maybe because to avoid more fraud?
well, that may have been, but that's what, I mean, but I think Visa, we all trust Visa, right? And our ATMs, and they're not using those kind of computer systems. They use a computer system that's fast and updated. So I think a lot of it is just laziness and waste. They didn't spend the money on buying new computers. They spent the money on, you know, putting gold toilets in weird places and, you know, building bridges that don't exist. I mean, they've done goofy things. But to get back to the original question, I think that there's a good chance that CODIS database, you would hope it's updated all the time, but that's something that probably doesn't get done as often as it would. And if you were somebody that was fighting for your life against a criminal charge with the government, you may want to have that database updated quite a bit. So I would assume it probably gets updated on a yearly basis like the database does for fingerprints, but that's a good question, Christina. Who knows? Any other questions before we wrap up, guys? Well, thank you for coming in. I'm sorry we ran so much longer. This time I'll try to keep tomorrow's session right at the 2-hour window. Thanks again for showing up, everybody. And you should be able to use the same links for tomorrow. And hopefully we'll see everyone back tomorrow. Thank you. Y'all have a nice day.
Thank you. Have a good day.
Okay, bye-bye.
Bye.